Perfect. So hi everyone, and thanks a lot for attending this month's session of the NeuroMeetup talk. So today our first speaker is going to be Samantha Weber from the Inselspital in Bern. So Samantha is currently finishing a PhD in the field of functional neurology at the University Hospital in Bern and at the University of Bern in Switzerland. She received a master's degree in health science and technology with a major in neuroscience from the ETH in Zürich. And throughout her studies, she worked on rodent models for neurodegenerative diseases. Eventually, she was awarded with the Munich Brain Course Scholarship from the Ludwig-Maximillian University in Munich, where she discovered her devotion to clinical neuroscience. She could then follow a new patient, starting a master's thesis in the Functional Neurological Disorder Lab under the supervision of Professor Selma Aybek at the Inselspital in Bern, where she had the opportunity to combine both her technical knowledge and a strong interest in clinical neuroscience. She's fascinated by the work with neuropsychiatric patients and by how few is known about their condition. And she then decided to deepen her knowledge by pursuing a PhD in the same lab. Her current research interest focuses on investigating various biological, neurological, and psychological diseases, markers, in functional neurological disorders. And recently, she returned from a research stay at the University of Santiago de Compostela in Spain, where she investigated gene-environment interactions under the supervision of Professor Ángel Carracedo. Today, she will present her latest work on functional neurological disorder, a neuropsychiatric condition at the border between neurology and psychiatry, in the framework of biopsychosocial stress model. So before I let you start, Samantha, I would like to remind the audience that you are free to ask any question you would like to transmit to the speaker directly into the chat. We will have 20 minutes of talk by the speaker, followed by circa 10 minutes of question and answer to answer these questions. So Samantha, the floor is yours.
So, well, thank you very much for this kind introduction. So then I start with functional neurological disorders, the contemporary nature of Charcot's dynamic lesion. So functional neurological disorders, or shortly FND. So probably not many of you have previously heard about this disorder, which is also normal, as sometimes not even neurologists are fully aware of it, and also how to diagnose it and how to treat it. So FND is defined as neurological symptoms that cannot be explained by an underlying classical neurological disease. These patients therefore can have a variety of different neurological symptoms, such as motor dysfunctions like paralysis or tremor, sensory dysfunctions like numbness, and also so-called non-epileptic seizures. These symptoms can appear and also disappear randomly and are also inconsistent with, let's say, classical neurological symptoms, such as, for example, observed in stroke, also multiple sclerosis. And therefore, clinicians also often perceive them or used to perceive them as faking by the patients. The diagnostic process is like very time and also cost consuming, as these patients often undergo multiple referrals with many different doctors. One reason or one problem here is also that there are no objective biomarkers available for FND. But let's go a little bit back in time. So FND is actually very old. Hippocrates was one of the first ones who reported on the symptoms, symptoms similar to those nowadays observed in FND. Hysteria, as he called the disease, was back then solely attributed to women, and he said it is caused, for example, due to malnutrition or also sexual frustration. In the late 19th century, then Jean-Martin Charcot, so the guy that you can see here in the picture, postulated that symptoms might be produced by a dynamic neurological lesion, which he said could be triggered by emotional trauma. Later on, then also Sigmund Freud assumed that patients must have suffered traumatic life events, which then converts the psychological distress into somatic symptoms, which then also gave rise to this term conversion disorder. So now with the upcoming of functional magnetic resonance imaging, so fMRI, in the late 20th century, intrinsic differences in brain function, so this dynamic lesion of Charcot, could then suddenly be studied, and also a new line of research developed to not only understand why, so in terms of preceding trauma, but also how the symptoms in FND develop. And Johannes and I would like to show you today a little bit about the current state of the art and also why we call it functional neurological disorders now. So studies using fMRI have provided a comprehensive evidence of multiple brain functional alterations and also network dysfunctions in FND. So in summary, key regions involve the insula, the amygdala, the hippocampus, also the temporoparietal junction, motor regions, and many more. So of particular interest, especially for today, is the insula, the amygdala, and the hippocampus. The insula represents an important hub of the salience and also limbic network and is associated also with conscious and unconscious emotion processing, so kind of emotional awareness, as well as with interoception, so the perception of the body state, also in relation to its emotional state. So in FND, then the insula has also been thought to be implicated in the mediation between emotional influence on motor control regions. Likewise, the amygdala and the hippocampus have been associated also with aberrant motor planning. So they are also very important key regions of the limbic system. And as such, it is also suggested that hippocampal, insular, and amygdala structures might in a way directly alter motor planning and execution in a way by bypassing also executive control regions. And this has then further also been thought to explain the development of functional neurological symptoms. So in summary, the question on how the symptoms develop has been extensively studied in terms of functional neuroimaging. So such the symptom production has been associated also with a large-scale brain network dysfunction involving sensory motor networks, interoceptive regions, and also abnormal emotion and stress processing. Now, for the question on why the symptoms develop, it has historically been associated with psychological stress or trauma, as you just have seen in the previous slides. So recent models, however, focus rather on the integration of a biopsychosocial model in which precipitating, perpetuating, and predisposing factors are combined more in an integrative stress diathesis model. So as such, childhood trauma, especially and unfortunately often sexual abuse, is still frequently associated with FND, but nowadays it's not a requirement anymore for the diagnosis. So moreover than chronic stress, also, for example, due to psychosocial factors like a misdiagnosis or also lack of treatment can enhance or also trigger the development of FND. Lastly, and also more recently, then research also focuses on the investigation on potential biological vulnerability factors in terms of, like, for example, a genetic predisposition, which can potentially make an individual less resilient to stress. So unfortunately, many of these, let's call them risk factors, have been studied relatively isolated. So in our study now, we therefore set out to investigate psychological and biological risk factor and their neural correlates in a joint manner, also to then shed light on these neuropathophysiological mechanisms in FND, and also to provide a better understanding of these fundamental properties of Charcot's dynamic lesion in patients with FND. So in a first study, we then aimed at investigating biopsychosocial vulnerability factors, which might explain the development of FND in a subgroup of vulnerable individuals. So to study psychological risk factors, we investigated different kinds of traumata in our cohort of almost 90 patients and almost 80 healthy controls. We measured across different subscales, including physical abuse, emotional abuse and neglect, as well as sexual abuse. Our patients reported significantly more severe and also longer emotional neglect, also in comparison to the healthy controls. So especially in terms of duration. So what happens when we experience chronic stress? Stress in general causes the activation of the so-called hypothalamus pituitary adrenal axis, so the HPA axis, which then further leads to the release of cortisol by the adrenal gland. This is also, so cortisol is also a common biomarker to assess stress. The stress response itself is also very strongly regulated by the hippocampus and the amygdala, also as a response to emotionally arousing stimuli. So current models in psychopathology nowadays suggest two potential mechanisms in the state of chronic stress. So first, prolonged exposure to cortisol can cause a maladaptive downregulation or kind of habituation to the stress response. Also due to the hippocampus, which then starts to tonically inhibit this stress axis, which leads to an overall lesser release of cortisol. On the other side, cortisol is also suspected to have a neurotoxic effect. So especially also in regions with high glucocorticoid receptor density, such as, for example, the prefrontal cortex, but also the hippocampus and the amygdala. So to now objectively assess stress in FND, we therefore studied cortisol as an end effector of the HPA axis, which has been found to be a good biomarker for stress, as well as cortical volumes, also to detect potential neurotoxic effects of chronic stress. So as such, we measured cortisol at 9 different time points throughout the day. 5 samples were collected in the morning, directly after waking up, and then each 15 minutes, in order to assess the so-called cortisol awakening response. So this natural peak of cortisol directly when you wake up. Furthermore, we took 4 more samples between 2 and 5 p.m. in order to assess the diurnal baseline cortisol levels. So doing so, as you can already see in this graph, we detected a flattened cortisol awakening response in patients with FND. So acting now on this assumption of a potential neurotoxic effect due to the prolonged exposure to cortisol in regions with high glucocorticoid receptor density, we looked at brain volumes in these regions. So we detected a reduced hippocampal, but also amygdala volume in our patients, again, compared to healthy controls. So patients reported on more severe trauma and also longer trauma. And we also identified a reduced cortisol awakening response, so a flattened cortisol awakening response. Additionally, also we detected reduced subcortical volumes in the hippocampus and the amygdala. To study now whether there is an association between these factors, we applied a multivariate partial least squares correlation analysis. This showed that indeed reduced cortisol awakening response was associated with increased severity, but also duration of trauma. This indeed suggests that there happens some sort of long-term adaptation or downregulation of the HPA axis, which then eventually is also supported by our data on the reduced cortisol awakening response. On the other hand, however, we did not find any association between the cortisol measures and the reduced hippocampal and amygdala volume. So this data suggests that psychological stress might act as a precipitating risk factors, whereas a reduced brain volume in the hippocampus and the amygdala might rather represent a biological predisposition for FND and is not necessarily the result of becoming ill. So for now, we assume that there might be some biological vulnerability factor. So in our case, in the form of a reduced limbic volume, which might increase susceptibility to stress. So therefore, in combination with, for example, childhood emotional neglect, vulnerable subjects might develop psychopathology. But how can this now be connected to functional abnormalities or this dynamic lesion as postulated by Charcot? So traditionally, one can assess functional connectivity in the brain using, for example, a functional connectivity approach, at which one just uses temporal correlations. As such, a time course is acquired in the MRI. Later on, we can then define specific regions or also networks in our imaging data and extract the temporal signal from those regions or networks of interest. By then calculating the temporal correlations between each of the networks or regions, we can get such a functional connectivity matrix, at which a high correlation coefficient, here indicated in red, is traditionally interpreted as increased functional connectivity between two brain regions or networks. Doing so now in FND and comparing it to healthy controls, we can see that here in this graph, when statistically comparing them, our FND patients have a lower functional connectivity between the salience and the limbic network. But first, let me explain this graph a little bit better. So we have here on the X and the Y axis, we have 17 resting state networks, and we just do simple multiple t-tests on those different connections. Those that are significant different between patients and control are marked here with this small asterisk. However, as we do multiple t-tests, we also have to correct for multiple comparisons, and this is here indicated with the white dot. So those connections that also survived correction for multiple comparisons, well, is actually just the one between salience and limbic network. Unfortunately, using such a static approach only summarizes the temporal correlation between spatially distinct brain regions or networks, also here assuming that their activity remains constant over time. The brain, however, is a very dynamic system, and it also fluctuates between different states also in order to adapt to intrinsic and also extrinsic stimuli. So therefore, to also better understand this Charcot's dynamic lesion in patients with FND, we must look also at the dynamic behavior of the brain regions across our time course, rather than just summarizing it in a static approach. So one possibility to do so is to use co-activation patterns. In our static approach, we identified the limbic and the salience network as being the most discriminative between patients and controls. Therefore, for a dynamic functional connectivity analysis, we therefore focused on the hubs, or like the centers of these networks. So in our case, the insula and the amygdala. Now, in a very first step, we only selected those images in which the insula or the amygdala are highly activated, so above a certain threshold. In a second step, then, we clustered the remaining volumes to receive cluster with similar co-activation patterns. So the result of such a co-activation pattern analysis is to have different clusters with brain co-activation, to which we can also, for example, refer to as brain states. So humans are now suggested to also constantly switch between different brain states, as previously mentioned, also to adapt to different stimuli. So this method, therefore, can help us to better characterize dynamic functional alterations in FND patients, which also might underlie this fluctuating behavior of the clinical presentation of their symptoms. So using the insula as a seed region, as you can see here on the left-hand side, we identified 3 characteristic brain states, at which the first one represents an insular co-deactivation with the default mode network. The second one, a co-activation with somatosensory regions. And the third one, a co-activation with regions that are involved in emotion regulation and interoception, such as, for example, the anterior cingulate cortex, the thalamus, but also, for example, the mesolimbic dopaminergic system. Then on the right-hand side, using the amygdala seed region, we identified also 3 different co-activation patterns. And the first one also represented an amygdala co-deactivation with the default mode network, as well as in the second one, a co-activation with the default mode network. The third state just represented a co-deactivation with the visual network. So further, when we then looked at temporal measures, such as, for example, duration of a brain state, so for how many time or for how long a patient or a healthy controls remains in a certain state, or also how many times a subject switched between different states, we detected significant differences between patients and controls in the amygdala and insular co-deactivation patterns with the default mode network, as well as the insular co-activation with the regions involved in emotion processing and interoception. So where do these functional abnormalities come from, and are they in a way associated with stress or even the clinical symptom presentation? So using again the multivariate correlation analysis, we identified a relationship between dynamic functional alterations and stress biomarkers, also again cortisol, as well as with their symptom severity. This also suggests a very important role also of acute stress on the fluctuating functional alterations, as well as of the functional neurological symptoms themselves. So in summary, we potentially identified a biological trait marker or vulnerability factor in terms of a reduced limbic brain volume. In combination with psychological risk factors, this might contribute also to the development of psychopathology. Moreover, then also this prolonged exposure to trauma might also lead to this maladaptive habituation or downregulation of the HPA axis, which is also supported by our data on the reduced cortisol awakening response. So then acute stress and stress biomarkers in general then also might stand in direct relationship to the dynamic functional alterations that we have observed in FNE patients. And those also might then further underlie the development of the functional neurological symptoms. So, however, as typical in clinical research, this data and also their interpretation really must be handled with care. So results have to be replicated and validated, and also longitudinal studies must be conducted in order to make further statements on causal relationships between potential predisposing and preceding risk factors in FND. So moreover, I also want to avoid to generalize our data and also to point out that individual biological, neurological, and psychological risk factors must not be seen like as a requirement for the diagnosis, but rather within the framework of a multifactorial model, so in which also others potentially not yet identified factors such as genetic or epigenetic markers might also play a key role. So with this, I'm already at the end of my presentation, and I really want to thank the organizers of the NeuroMeetup for having us here and for this excellent organization. I think it's a really great initiative, and I'm happy Johannes and I have the possibility to present here today. So yeah, of course, thanks also to everybody who came and is listening. And a special thanks also, of course, goes to Professor Selma Aybek and her team and all our collaborators. So also, if you're interested in our research, you can find us on the Clinical Neuroscience Bern webpage, or you can also follow us on Twitter. So thank you very much.
Thanks a lot, Samantha. Very interesting talks and very exciting results.
Thank you.
Yeah, congratulations. So I would like to remind people in the audience that they are free to ask any question they might have directly into the chat. So that I will transmit them to the speaker after the talk. Maybe I will start with very quick naive questions. You mentioned at the beginning of the talk that FMD was not very well known disorder. Do you have numbers about the prevalence in the population?
Yeah, it's very often. So I think there are studies that show that every third patient in neurology actually has some functional disorders as well. I guess the problem here is as well as very often they also overlap, for example, with other like structural diseases. And therefore it's like kind of also difficult to sometimes detect what actually is like an organic damage and what is actually functional. But yeah, very often clinicians are just not really aware of it. So it exists, but it's often like difficult for the patients or like a long journey until they actually get to the correct clinician to make the proper diagnosis and to provide them with the correct treatment. So it's not that it's rare, but it's like still like kind of a stigma there. Yeah.
Makes sense. Okay. Thanks. We have a question from Johannes. Did you look for subgroups within the overall group of FND patients?
Well, not fully. So we actually kind of have this transdiagnostic approach that we just took any kind of symptoms that we could gather. But we did, in a way, some subgroup analysis more on the biological level. So, for example, only in females, and so on. So, well, we corrected the data, like the cortisol and also the structural data for common confounding factors like age, gender, also like the cortisol for a lot of like, for example, menstrual cycle and all these factors, but still we wanted to also look into these subgroups, but more actually on a on a biological level and not really on a clinical level. But unfortunately, we also do not really have like patients with really clear symptoms in a way that we can say we only look at seizures or we only look at motor function or motor dysfunctions. So we have patients with many overlapping symptoms, which could also make the subgroup analysis a bit tricky in that regard. Yeah.
Thanks. We have a new question from David Tanner. Is FND officially classified by the World Health Organization?
Yes, it is. Well, it's changed its name very often, but actually, yeah, you can find it in the DSM-5, and I think, I'm not fully sure, but I think not yet in the ICD-11. Yeah. Okay.
Another question still from David Tanner. Do you have any epidemiological data on FND? Sorry. For example, did FND increase over the last centuries, or are FND more common in countries with higher stress level?
Well, I need to like freestyle here a bit, but there is recently also a lot of research going on in that direction, for example, with COVID, because there was like the lockdown and everything. So I don't know, maybe Johannes, you know more, but I think there is like a higher incidence now also due to the lockdown and due to COVID. But I think it's like still a bit like contradictory, the results there and the research in general. But yeah, I don't know, Johannes, do you know anything else for that?
Yeah, I think it's a great question. I think looking at historical data, it's really hard to say, because I think FND kind of got lost throughout the 20th century. So there was, as Samantha introduced, like this Freudian time where it was really big, but kind of when Freud wasn't that huge in psychology anymore, conversion disorder kind of got lost, and it was only rediscovered in the 1990s or in the early 2000s. So it's hard to say. And the same is true for cross-cultural comparisons, because from what we know in cultures different from Western cultures, it is just as common, but it tends to have kind of cultural shape. So expectations and symptom expectations seem to play a role. So it's really hard to define what is what in which context. But I think overall it's about the same across cultures, just looking at the numbers. But what Samantha just introduced, the idea of stress related to COVID, but also the symptoms that people endure, and this really changes over time. So related to stress, yes, you can see that quite clearly, also in more closely monitored populations. Yeah.
Thank you very much. Also, Samantha, for the great talk.
Thank you.
All right, so thanks a lot for answering the questions. So thanks again, Samantha, for your talk. I guess we can now move on to the second speaker, Johannes, if you are ready. I'm just going to quickly introduce you. So Dr. Johannes Jungiligans is a neuropsychologist and affective neuroscience researcher focusing on neuropsychological and affective aspects of functional neurological disorder, which is a common neuropsychiatric illness. He is currently a postdoc at the Department of Neurology at the University Hospital Knappschaft Bochum in Germany. After getting his PhD at the Ruhr University in Bochum, he was a visiting postdoc in the lab of Professor David Perez at the Harvard Medical School. And in his work as a clinician researcher, he investigates the neurobiological underpinnings of emotions, interoception, and metacognition in patients with functional neurological disorder using multimodal assessments. So Johannes, the floor is yours.
Well, thank you very much. Once again, also from my side, thank you for organizing this really interesting forum here, and thank you for having me here. My talk really has a long title, and it basically contains everything that I'm going to talk about. So it's called Linking Gene Expression Patterns and Brain Morphometry to Trauma and Symptom Severity in Patients with Functional Seizures. And just from these buzzwords here in the title, I guess you can see the connection to Samantha's great talk. So it's really well fitting to that. But first, let me paint a bit of a bigger picture here. We know, as Samantha also introduced, that adverse life experiences play a huge role in functional neurological disorder, but also play a huge role whenever they occur. So they tend to heavily influence the pathway that the life of those affected by those traumatic experiences take after those experiences. So specifically, they increase the risk to develop any kind of psychopathology later in life. And we do know that for those who do go on to develop a psychopathology, that the burden of the trauma is really associated with an earlier illness onset, a greater symptom severity, and a more treatment refractory course. So really a more severe burden of illness across a range of psychopathologies. And in our understanding, these effects of these traumatic experiences, which mostly really happen more early in life than or earlier in life than the symptom onset, which mostly then happens maybe 10, 12, even 15 years later, that these effects are moderated by how the brain maturates across along the time, and that there are certain effects of trauma on the neuroplasticity of how the brain develops, and that specifically different kinds of trauma actually have different kinds of effects on how the brain maturates and develops afterwards. So we know, for example, that verbal abuse tends to influence the brain maturation, both in structural and functional networks, different from how, let's say, sexual abuse influences them. So with that being said, let's turn over to functional neurological disorder and one specific subtype, that is functional seizures. So functional seizures kind of look like that. So you could easily mistake them for an epileptic seizure if you're not an expert. And as our other functional neurological disorders, they are not related to an underlying pathology of the brain. So it's not epilepsy, but it's one of those neuropsychiatric disorders. And we know that in those patients with functional seizures, there are elevated rates of adverse life experiences. So there has been a great Lancet psychiatry review by Lea Ludwig and colleagues, where we could see that at a ratio of about 3.1 compared to healthy controls, they suffer from a range of adverse life experiences, including emotional neglect, sexual abuse, physical abuse. And in FND, and especially in functional seizures, there seems to be kind of a dose-dependent relationship. So the more severe the trauma was, the more severe the patients tend to suffer from functional seizures. And also, not only the symptom severity itself is affected, but also the magnitude of adverse life experiences is correlated with relevant neuropsychological and psychiatric factors, such as the tendency to dissociate, which is one core feature of FND, but also features like emotion regulation and other, well, psychological and psychiatric features. So this leads to our understanding that adverse life experience actually maybe even have a mechanistic relevance in the pathophysiology of FND and functional seizures especially. However, what we don't know, or at least state of the art are not really sure of, is whether there is also a relationship between certain trauma subtypes and the symptoms endured later in life, and whether this relationship is moderated by developmental neurobiological processes, including genetic factors. So really, the overarching question that we're asking here is, why do these patients develop functional seizures based on that they experience something traumatic earlier in life? And here, I'm going to present a neat little research project that we did in a rather small cohort of patients, 20 patients with functional seizures who were all neurologically healthy. 15 female to 5 male, which is kind of a typical gender ratio. And those patients underwent the traumatic experiences checklist, which is a questionnaire that measures the extent of emotional neglect and abuse, physical abuse, threat to life, and sexual trauma that those patients endured at some time point in life. And the patients also filled out the somatoform dissociation questionnaire, which is a questionnaire on functional symptom severity. So we had data on both the potentially predisposing traumatic experiences and the symptom severity as of the time point we're measuring. When we looked at the relationship between trauma burden and illness severity, we found that for some kinds of trauma, specifically for emotional neglect, threat to life, and sexual trauma, there really was a significant correlation. The higher the trauma burden, the more severe the symptoms were. Now, as you remember, we wanted to understand how the brain, the biological basis, plays a role in this relationship. And we did this by doing a gray matter analysis, not actually by looking at those clinical MRIs, but by doing an analysis type called voxel-based morphometry, which is one way of looking at the gray matter volume, the cortical volume of those patients. And what you do is that you basically take all those MRIs of those patients, stack them on top of each other, do the same with questionnaire scores, and then you can kind of check within this whole group which of those voxels, so these tiny 3D pixels that make up an NMR image, are related to, let's say, those questionnaire scores, and whether or not the volume of the cortex in a certain area is related to the extent of trauma or the symptom severity. When we first look at the results related to the symptom severity scores, we found that the higher the symptom scores were, the lower the cortical volumes of a range of areas was, including the bilateral insula, so here, right, left and right, but also the right amygdala, the orbitofrontal, and the perigenual and posterior cingulate cortex. So a network of regions in which the volume, the cortical volume of the gray matter was related to symptom severity. We did the same kind of analysis in the trauma subtypes and found that for emotional neglect trauma, once again, the left and right insula and the right amygdala were related inversely, so a negative relationship once again to the extent of emotional neglect endured earlier in life. So the higher this emotional neglect burden was, the lower the volume of those bilateral insula and amygdala regions. For sexual trauma, we once again identified the right insula as being related negatively to trauma burden, also the right putamen, so right down here, but also the right cuneus, so this posterior parietal occipital region here, and you might see something similar here, and actually that is kind of a common finding across studies related to trauma, that posterior regions of the parietal cortex and occipital regions have a higher volume. So we find that here too. But I think most interestingly is that those networks that were affected, mostly surrounding the bilateral insula and the right amygdala, really neatly overlap with what we found earlier in the symptom severity analysis. So next, we wanted to understand how this might unfold throughout lifetime. So what factors might play a role in moderating this relationship? And we turned to a method called neuroimaging genetics, which is kind of a novel method that I want to introduce here because I think it's really a cool tool in case you don't have individual genotyping data, which we didn't. And neuroimaging genetics really allows you to get at least a glimpse at what genes or genetic factors might be influencing the neuroimaging findings that you are identifying in a given cohort. So this is what we want to get at, which looks kind of messy and a little weird. So let's break this down step by step, really. So First, what we did was that we took those gray matter findings. So here are those related to symptom severity, and we related those to an atlas of gene expression data. This is a huge project by the Allen Institute in Seattle, the Allen Human Brain Atlas. And this atlas really is a map of which genes are expressed in the brain. And they developed this atlas by taking post-mortem brain tissue data from a huge number of places throughout the brain, and then checked which genes were expressed in those tissue samples, and were then able to map which genes are expressed where in the brain. And they did this across a range of post-mortem brain samples. And so we kind of have a really good idea and a map of which genes are expressed where in the brain. And now what we did was a spatial correlation. So we correlated our neuroimaging finding with this gene expression atlas, which then allows you inference. So it tells you something about which genes are overexpressed in the implicated brain regions. So which genes are overexpressed here in those blue, light blue regions, and without the need for individual genotyping in an individual patient. And in this case, you get kind of a distribution of which genes are expressed there and how similar they are. And we took those genes with the highest similarity, and then you really get a list of genes which have the highest similarity to the expression patterns, a list of genes whose expression patterns really highly correlates with those neuroimaging findings. And in the case of the genes related to symptom severity, we identified 67 genes. And then we did the exact same analysis for the maps related to emotional neglect and to sexual trauma. And each time got a list of a number of genes, 17 in emotional neglect, 36 in sexual trauma. And so we had 3 lists of genes. And now you remember we wanted to understand how these might map onto each other. So what we did was really that we combined those lists and looked what genes overlapped between those lists and identified those which appeared basically in both lists between symptom severity and emotional neglect, but also for symptom severity and sexual trauma. So now we really ended up with 2 lists that contains first list, which contained the overlap between symptom severity related genes and emotional neglect related genes. You can see here a bit tiny, I guess it was only 3 genes overlapping between these 2, but overlapping between symptom severity and sexual trauma related genes, there was a list of 22 genes. Now, I'm a psychologist. I don't understand anything about genes except that they are kind of important. But if you just show me a list of genes, I have no idea what they do. But it turns out that biologists, especially geneticists, seem to be really great people because they just put everything they know about genes online into huge databases. And then dumb psychologists as I am can go there and input a list of genes. And these databases just tell you what functions, what things are those genes related to. This technique is called gene set enrichment. So it really searches for the functional, structural, and process annotations, so the meanings of those genes that are statistically overrepresented in a given set of genes. So for example, those 22 genes here, to tell psychologists like me what those genes do. And so we did exactly that. And for those 3 genes, we didn't find anything. Maybe it was just not enough. But actually for those 22 genes overlapping between symptom severity and sexual trauma, we found that those were related to a set of receptor signaling pathways, specifically to adrenergic receptor signaling, to serotonin signaling, to opioid signaling, and GABA receptor signaling, as well as to enkephalin. So a range of mostly stress-related neurotransmitter systems, which is obviously very interesting when we're talking about neuropsychiatric illness, also looking from a psychopharmacological standpoint. So really an interesting finding that those genes are overexpressed in those regions. Next, we wanted to understand the neurodevelopmental implications. So we did another gene set enrichment analysis. So input the same list of genes into another analysis tool, which explains or shows how those genes are related to neurodevelopment. And we found that those 22 genes were actually highly implicated in cortical development from neonatal or early infancy all the way through young adulthood, and related to amygdala development from adolescence and young adulthood. So two time periods where we understand that especially there, adverse life experiences tend to have a huge impact on how those patients develop later in life. So this is what brings us to this rather complicated and a bit messy figure here. So we're starting really from brain structure alterations, going all the way to which genes and which cortical regions are implicated there. For those who are interested in the nitty-gritty of this project, please feel free to check out this paper, which also explains the details of the methods here. It was recently published in Psychiatry Research Neuroimaging. And I want to spend the last 2 minutes of this talk to talk a bit about the implications of these findings. Admittedly, small group and really indirect way of measuring what you're looking at, but actually this fits rather well with the small but growing body of literature on genetic factors in FND. And I really want to highlight one of those studies done in Selma Aybek's group by Kalliopi Apazolu, who identified an increased methylation of oxytocin receptor genes. So once again, a stress-related signaling mechanism. And this from Mark Hallett's group, which is really a huge FND research group in the United States, who also found serotonin receptor-related alterations. And next, and this is where we really tie in with Samantha's talk, I want to talk about the networks implicated, which showed these structural correlations with symptom severity, because as you might have identified, those map really nicely onto those two overarching brain networks that Samantha also introduced, the default mode network with those frontal, orbitofrontal, and posterior cingulate cuneus alterations, and the salience network with the bilateral insula, the anterior cingulate cortex, and a little bit lower also the amygdala, which is kind of interesting because those networks actually have huge implications for predictive processing frameworks. And predictive processing frameworks are really powerful when you want to talk about how specific FND symptoms emerge. And we recently thought a bit about this and the role of emotions in this whole picture in a paper cited here, and especially the role of the insula in interoception, so the brain's moment-to-moment modeling of what is going on in the body, which is understood to be highly important for FND, as you might understand rather intuitively, that knowing how stuff goes on in the body really influences how you feel in your body and maybe what's going awry. So this really is it. Thank you so much for your kind attention. Thank you so much for your interest and for having me. And I'm really looking forward to the discussion. So thanks.
Thanks a lot, Johannes. Very cool talk again. So yeah, those are two very exciting results. So please feel free to ask any question you might have in the chat. We have a first question from Samantha. Yeah, I had the same question actually. Did you also analyze the overlap between emotional neglect and sexual trauma?
That's a good question. Actually, we didn't. Would be interesting. I don't know why we didn't. We were really specifically interested in basically the mapping of those predisposing traumatic factors to how those symptoms play out later in life. So no, we didn't. I could go back to the data and check that actually.
Okay. There is a follow-up question from Samantha. How can we, maybe in just 2 or 3 sentences, then link emotion to interception or interoception?
Okay, well, that is a great question. And I'm not sure if that's a 2 to 3 sentences answer available in my head right now. So I think that really interoception is much more than we consciously know about ourselves. So it's not just the way our hearts beat or the way, I don't know, you feel inside, but it's really all the information the brain has about the state of the body, glucose levels, temperature, sweating, whatever, you name it. And that those informations are really important for how the brain perceives what is important, because the brain's main job is to keep you alive, to keep your body in basically a good shape, to have enough energy. And that so it also influences, so the state of the body influences how you perceive information, how you perceive what is important in life or in the world around you. And that includes what situations might elicit an emotion, or what kinds of emotions are there, or how you experience emotions. So I think the state of the body informs your predictions of how you perceive and generate information, including emotions. I think that's the shortest kind of answer I can say to that.
Thanks a lot. I might have a very short and maybe a bit naive question about the methodology. If you can share again your presentation.
Yeah, sure.
And go to the slide about the neuroimaging genetics.
Yeah, give me a second here.
Yeah, sure.
Oh, sorry. I'll be right with you.
No problem.
So there we are. And we're going, why does it always happen? Ah, man, give me, okay, so now we're talking. Here we go.
Okay, awesome. So maybe there is something that I missed, but in the middle, you compute the number of genes that overlap between symptom severity and sexual trauma.
Yeah.
How come? When you say n is equal to 14, this is the pool of genes in sexual trauma that do not overlap with the symptom severity.
Right. So
okay, yeah.
Right. So basically, what you can see in this overlapping area is really those which overlap, and the rest. So the n like equals 45, and the n equals 14 are those which do not overlap.
Okay. No, that I didn't get that. Okay, perfect. Thanks a lot. Makes sense now.
Yeah, sure.
All right. Do we have any more questions from the audience? Last chance. All right, looks like we don't. So I think we're coming to an end now. So I would like to, no, this is just thank you. Will you post a recording of the session, please? Also for future reference, could you? So we are definitely going to post a recording of the session. And as a matter of fact, I'm going to give you a link to the YouTube channel of the NeuroMeetup. Yeah, you go. So yeah, we record every session and they are freely available on our YouTube channel. So yes. All right, so if there is no follow-up question, I would like to thank everyone in the audience for the participation, the attendance, and the nice questions, and especially to thank Samantha and Johannes for for participating. Those are very exciting and interesting results. And I guess I'm speaking in the name of everyone to say that we are really looking forward to the future results.
Thank you so much. Have a great evening, everyone.
Thanks a lot. Thanks, Samantha. Thanks, Johannes. And thanks to everyone. Thanks a lot. Bye.